Welcome, everyone. Thank you so much for joining us. Today, we actually have a very, very great guest, John Amrhein. He is Vice President of McDougall Scientific, very leading edge CRO, niche CRO here in Canada, primarily focused on statistics and data management. John's has advanced from a frontline statistician through increasing levels of responsibility to his current position as second in charge of McDougall Scientific. In each position, he has made a positive impact on the organization and its customers' business processes by providing evidence-based decision support or by instilling in others the know-how to make decisions using their operational or research data. So it gives us great pleasure to have John as our guest and talk to us about novel clinical trial designs using statistical methods. So with that, I'm going to turn it over to John, and we'll go from there.
Thank you, Chris. Hadn't heard that introduction in a while. It's nice to hear that again.
You're welcome.
And good morning, afternoon, or evening, everyone. I want to thank Chris and Heather for inviting me to take up one of their slots in their webinar series. It is a pleasure to be part of this webinar series. My presentation today is a description on how you might accelerate your clinical trials, and accelerate meaning to get them conducted in a shorter period of time, let's say, through innovative trial designs. There's a lot of innovation that's happening in clinical trial design space right now. And I'm going to take a little detour with an introduction to Bayesian thinking. I hope that at least some of you saw this subtitle and decided that was reason enough to attend this webinar. A lot of the innovative designs these days rely on this idea of Bayesian statistics or Bayesian methods. My objective really is to either convince you or to encourage you to consider engaging a statistician early in your study design process. And it could be a clinical trial, could be some other type of study, a registry study or so forth. And a statistician's job is to make your study design efficient. And we'll talk about that throughout the presentation, how we might do that. A pictorial of our agenda, I'll first describe clinical trial workflow and how you might consider, or how you might think about the workflow of a clinical trial, and then counter that with how a statistician thinks about the clinical trial workflow during the design stage of your clinical trial. That will take me into a description of what role does a statistician play on the team that is designing your study. It's at that point we'll cross the street and take a little detour on what is Bayesian thinking, and I'll come back to that when we talk about the innovative designs that are available and that you might consider. These, the short list here on this slide is adaptive designs, something called basket and umbrella designs. You may have heard of platform trials. You might have heard the ability to use previous data, historical data, literature data, so forth. So we'll talk about some of those ideas.
Can you share your screen?
I'm sorry.
That's okay.
All right. Well, we haven't missed too much.
Earlier, just a little refresh, and I think we're good to go.
Can I share my screen? I don't know, Chris. I'm not sure if I can or not.
There we go.
You're seeing it now?
Excellent.
Okay, now I got to move you out of the way. All right, so here's what you missed. You missed this great skyline of the Toronto, where we are located, my title slide, my objective, which I read off to you. And my marketing guy would not be happy. He did a lot of work on these slides. And then this is our agenda. All right.
Perfect.
So that brings us up to date. Apologies, Chris. Before we get into that, who is talking to you from McDougall Scientific? John Amrhein. Yes, as Chris said, I'm vice president. I have 3 major stages in my career. I worked for U.S. Department of Agriculture for about 10 years. I learned all about probabilistic methods in statistics, conducting surveys. I find what I learned at that stage of my career is now creeping into what we call real world evidence with weighted estimates and so forth. Then I went to work at SAS Institute, the software company, where I taught statistics, I taught programming. I was a consultant in different spaces, including life sciences, but also in manufacturing, finance, insurance, government, telecommunications, and so forth. And that's where I really solidified my knowledge of statistical analyses. And then I joined McDougall about 13 years ago as a senior statistician, took on multiple hats now, and eventually winded up where I am right now. And I was thinking maybe 13 years sounds like a long time, but let's remember that cicadas have been sleeping that entire period. McDougall Scientific is a niche CRO in Toronto, Canada. Niche because we concentrate on data-related services, that is data management, statistics, and then related technology. This arch in the slide that you're seeing lists our areas of services. The two we're going to be concerned about are in the lower left. That's the clinical trial design, which is part of our statistical strategizing with our clients and consulting. I will point out over on the right that we do provide technology often, and most often really, that's in partnership with organizations like TrialStat, which is what has brought us together today. We support all phases of studies, 1, 2, 3, and 4. You can see, based on this histogram, our sweet spot are phase 2 and then phase 3. We're getting more phase 4, especially in Canada with our large pharma clients, but we also support device, diagnostic, preclinical, some surveys, and then there's a smattering of things that don't fit into any of those categories, including one project where we were asked to forecast stock prices. Therapeutic areas across the spectrum. Recently, we've been doing a lot of oncology studies, which attracts more sponsors in that space. So that's our primary area of focus. We've done a lot of pain studies, device diagnostics for a period, but we've hit quite a few different areas. So we do not specialize in any one area. And I rather enjoy working in the different therapeutic areas because we often borrow ideas from one therapeutic area into another. Okay, so first let's talk about the study process. This is as a clinician might see it, and you're used to this. This is the data flow. We recruit patients. You record the data about those patients at the sites, and then individuals in data management or the CRAs do their source data verification, the query process, clean the data, make sure every patient's data tells a viable story. Then the statisticians will get busy after database lock to analyze the clinical data. And finally, a report is generated, maybe just in tables, listings, figures, but maybe a clinical study report, maybe a publication, and that's the end result. Statisticians, especially at the design phase, reverse this process. And this is to design a study. We first talk about or think about the clinical study report, and we will ask our clients, what is it that you want to write in your CSR or in your publication? And it might be, we ask for specifics. It might be something like our pain medication reduced the pain score 20%, whereas in a placebo, the effect was a 5% pain reduction. That tells us a lot about what it is you want to do. It helps us identify which analyses need to be performed, whether it's going to be longitudinal data or not. So we consider then analysis options that will be needed to answer that research question or allow you to write that statement in a CSR or a publication. Once we know which analyses we're going to perform, that tells us which data will be required. For example, if it's you're looking for answers to your question at different time points, then we need to conduct repeated measures. We need to collect those data at different time points across the study duration for an individual patient. It's only after that that we can start considering the different design options. And we'll go through quite a few different design options. But the statistician's job is to make your study as efficient as possible. Efficiency we'll talk about. That's reducing the number of patients required or reducing the time required. And only after we have narrowed the design options to either one or maybe 2 or 3, then we can conduct your power analysis, determine the sample size, and that might tell us which of the short list of design options you want to use. That's the statistician's view of the world. We start from the endpoint and we work backwards. And what's our role in the team and in coming up with your study design? There's several aspects that go into our role. And these portraits on the screen are meant to convey those different aspects that make up the statistician's role in designing a study. And the first quote is by John Tukey, an American statistician. And he famously said, well, it's famous among statisticians that the best thing about being a statistician is you get to play in everyone's backyard. And what he means by that is that statistics is applicable to many, many different areas. I mentioned that when I worked at SAS, I worked in manufacturing, telecom, retail, finance, insurance, government, so forth. That's a lot of backyards to work in. And the statistical methods that work in one backyard work in the other backyards as well. Now that I am at McDougall and working in the life science clinical research, the backyards have changed. The different backyards are maybe drugs versus devices or diagnostic devices, but also the different therapeutic areas. And I'm often asked, well, does McDougall have experience in, and then name a therapeutic area? And my view of the matter is you should be asking, does McDougall have experience, and then name a statistical method? Competing risks in survival analysis. That's what's important. We can take the statistical methods and apply it to the different therapeutic areas. And we do that by relying on the other members of your study design team. And that brings us to Helen Keller's quote, alone we could do so little, together we can do so much. And I'm going to go through in the next slide the different members of the team and the role that each of them plays and how the statistician interacts with them. If you don't bring a statistician onto your team or don't do so early enough, then the statistician might find himself or herself in the situation that Homer Simpson finds himself on the screen. It was like that when I got here. And I say that because I sometimes receive a protocol that has been approved. Maybe the trial has already started. As we review the protocol, we find opportunities where things could have been done a bit more efficiently. Maybe a different analysis, maybe fewer patients, or maybe we could have done a little adaptation in the midstream. But it was like that when we got here. So we do the best we can with what we are presented. That's the situation you don't want to find yourself. And here is the protocol team. I'll leave the statistician alone for now. Let's start with the regulatory affairs. The statistician works very closely with regulatory affairs. Regulatory affairs maps out the regulatory strategy. What will the regulatory authority, FDA or Health Canada, require of your study in order to move to the next step, the next phase of study, or maybe for market approval? That's the regulatory pathway or regulatory strategy. So that's what will be required of the study. The scientist and the product expert together know your product, its mechanism of action, know how it works, and can provide valuable insight to the statistician on some nuances of what variability we might see in the outcome that's being studied, what data it's possible to collect or when it's going to be needed, so forth. The medical doctor knows the target population, knows the patients, knows what they're experiencing, knows what might be practical and what might not be acceptable in your clinical study. For example, in oncology, I've come up against a regulatory strategist saying the FDA requires biopsies at certain time points. And the medical doctor counters that that's not acceptable. No oncologist is going to subject their patients to that many biopsies. So either recruitment is not going to happen, it will be very slow, or you won't be able to contract the number of sites that you need. So the medical doctor plays a very key role in what data can be collected from the patient population and what might be seen as in terms of outcomes. Finally, the clinical operations team weighs in on what's practical, whether it's practical or logistically possible to collect the data that might be required. So how does the statistician relate to this? Well, it's primarily with the regulatory affairs. And the regulatory affairs maps out that pathway. And they say, well, I want to go to Paris, which means this is what will be required to satisfy the FDA. The statistician says, okay, in order to get there as efficient as possible, we're going to take a private Learjet, which means we're going to have some very complex, innovative design with a lot of requirements. And the sponsor weighs in, the business, what do I want to say? The CEO, CFO comes in and says, nope, can't do that. That's too expensive. So the statistician is meant to make your study efficient, how we're going to satisfy the regulatory requirements subject to constraints that come from the sponsor, either budget or the clinical ops with the practicality, or with the medical doctor suggesting what can or cannot be required of the patient population. Now I've mentioned the phrase, make your design as efficient as possible. Statisticians measure efficiency in terms of variability of the outcome. So on this slide, the mean is your outcomes average. Maybe it's a pain score, maybe it's months of survival, maybe it's the wound closure and wound healing study. Whatever your outcome of interest, and you're measuring the average, the variability is how likely an individual's outcome is going to vary from that average. The green line shows high variability, that it's quite likely that an individual's outcome, their pain score, is going to deviate from the average quite far and with fairly high probability. So with high variability, it's going to be difficult to get a confident estimate of that average or mean. The blue line is somewhere in the middle, looks like the famous bell-shaped curve or normal curve. That's medium variability. Low variability is what we're after. In the red curve, it's highly likely that an individual's outcome will vary only a little from the population average. And a statistician's job is to design your study such that the estimate that we produce at the end of the study is going to have confidence intervals or distribution closer to what we see with the red curve than that we see with the blue or the green. Okay, let's take a little sidestep here and describe what is meant by Bayesian thinking. You may have heard of Bayesian statistics, Bayesian methods. I use the phrase Bayesian thinking. It's not quite philosophy, but it's in that direction. Bayesians talk about a degree of belief and this is how we interpret probabilities. Let's take a simple example with the weather forecast. And each day you might look at the weather forecast and read what is the probability of precipitation, the POP. And you decide, you just based on personal experience. If the POP is 50 or less, then you don't take an umbrella. If it's higher than that, then maybe you take an umbrella with you or wear a rain jacket. But let's suppose that you have successive days, 4 or 5 days, that the probability of precipitation is 45%. You don't take an umbrella and you get wet 2 or 3 of those days out of that sequence. Well, now you might adjust your degree of belief of what the probability of precipitation means. And you might decide 40% is your threshold at which you think it's likely that it's going to rain. So what I just described is you had a prior belief about the probability of precipitation. You gathered some data, you have new experience, and you updated your prior belief to a posterior belief. This also comes into play in assessing risk. I have on the screen a parking ticket. Let's pretend that you park your car without buying the temporary parking ticket in some location in your neighborhood because you know the probability that you're going to get caught and have to pay a fine is so low, you're willing to take that chance. So you have a prior belief about your risk of getting a parking ticket. And then you read in the newspaper, you hear on the news that the city is conducting a crackdown on scofflaws, not buying these temporary passes. And you think, okay, well, now the risk is going to be higher. So instead of parking without buying that temporary pass, you now decide to buy it. So again, you had a prior belief about the risk of some event happening. You gathered some new information about this crackdown on scofflaws, and you adjusted your behavior accordingly. Bayesian statistics comes into play quite heavily in assessing cause and effect, or probable cause. On the screen, I have a little chart that I got off the internet about tuberculosis and the symptoms. Symptoms are the effect of an underlying illness, tuberculosis. So we have on the screen weight loss, fever, night sweats, a lingering cough, loss of appetite, tiredness. These are all symptoms of tuberculosis. They are manifestations of the underlying cause, tuberculosis. We can't really see tuberculosis. We just see these symptoms. You could also think about COVID, now that we're in the midst of a pandemic, loss of taste, loss of smell, fatigue, muscle aches, sneezing, coughing, all symptoms of COVID-19. But just because you sneeze or have a cough, does that mean you have COVID? Just because you're tired, does it mean you have tuberculosis? It's easy to estimate once we know the underlying cause, either COVID-19 for your cough or sneezing, or tuberculosis for the night sweats. It's easy to estimate that you're going to have one of these symptoms. But the reverse is rather very difficult. If you're just tired or you just sneeze, does it mean you have tuberculosis or COVID-19? Not necessarily so. But through Bayesian analyses, where we measure the effects and then estimate the probable cause, we're able to estimate with confidence when we see combinations of these symptoms what that underlying cause is. And the same with the COVID-19. Several weeks ago, I was watery eyes and sneezing and coughing once in a while. And I thought, well, this is allergies. It's not COVID. It happens every time this time of year with the pollen and trees coming out. But then I learned, my neighbor leans over the fence and says, John, we've tested positive for COVID. And I live in the city and our homes are right next to each other. I thought, okay, well, maybe these symptoms aren't allergies. So our entire household went and got tested. Luckily, it turns out that it was just allergies. But Bayesian thinking, Bayesian analysis is key for cause and effect. When you just see the effect, and you want to estimate probability of an underlying cause. And this was the initial purpose or motivation for Bayesian thinking, Bayesian stats. Back in the days of the Reverend Thomas Bayes and the philosopher and mathematician David Hume, instead of writing blogs back then, they wrote pamphlets, and David Hume published a pamphlet that said you cannot declare cause and effect unless you have data, empirical observation of that cause and effect. Before I continue, I'll say that this is directly attributed to what we are doing in clinical trials, because we are trying to determine whether our treatment causes the effect that we wish to see. So this is directly applicable. Now, back in the days of these gentlemen, David Hume's pamphlet made the Reverend and others like him uncomfortable because back then in the religious circles, there was what was called first cause. And by first cause in capital letters, they were referring to God. So David Hume was attacking the existence of God in their eyes. And the Reverend Thomas Bayes decided to counter David Hume's claim mathematically. And he figured out a little experiment to estimate the cause just from observing the effects. And an example in their world was the rising sun. What causes the sun to rise every day? In their mind, it was first cause, or God. But David Hume said, you can't say that unless you see God rising the sun every morning. So that's where Thomas Bayes came in and mathematically showed how you could estimate cause from observing effects. And he didn't publish his results. They were only published after he passed away. I'm not sure why he didn't publish them. But Bayesian thinking is attributed to the Reverend Thomas Bayes' early work. Okay, so what does that have to do with what we're here about, the clinical trials and trial design? Well, let's think about your phases of study. And let's pretend that you're designing a phase 2 study. On this slide, we see a solid blue line, and that solid blue line is what you know about your outcome and its distribution, pain score, survival, wound closing, whatever it is that you're interested in. Its mean and how much it varies is represented by that solid blue. And you use that indirectly now in our world of non-Bayesian statistics to do your power analysis and decide what sample size you need for your phase 2. In Bayesian statistics, that is our prior information. In our phase 2 study, which is the dashed line, the large dashed line, after the phase 2 study is over, maybe that's what our distribution looks like. And in Bayesian statistics, we take that information, that's the new information or new experience, we update the prior, the solid blue, and we end up with the dotted blue line in the middle. So just as I said with the parking ticket and the, what was the other example? The weather example, carrying an umbrella, prior belief, new information updated to a new estimate, a posterior estimate. And then if we are starting a phase 3, we would use the dotted distribution to plan our phase 3 study. Okay, Bayesian systems have creeped into our world. Some of you, well, all of you have one in your hands. Some of you might be looking at it right now. There is a Bayesian estimation system called Bayesian Networks, which is in your smartphone. It separates true signal from interference. It's at the heart of artificial intelligence. This idea of updating information with new data is how artificial intelligence is intelligent. It learns as new data comes in. Let's think about a pharmacovigilance program. Maybe some of you are at a company that FDA required you do a REMS program to get on the market. So you're collecting data as it comes in, looking for serious adverse events. You would start off with your prior information is what you did in your phase 2 and 3 studies, but you would collect new information and update the old every time new information comes in that you collect through your PV program. As this goes on long term, you would have very solid estimates of the probability that your treatment is a cause of a serious adverse event. So you could either get an early detection system and head off the use of your product in certain situations, if that's what's called for, or you would have evidence that your product is not the cause of these serious adverse events. Okay, let's get into the innovative designs. You've probably all heard about adaptive by design. We're going to, the next few slides are going to lead up to a study that we designed some years ago that has all of the elements that I'm about to show you. Some of these are traditional design features like stratified designs. I have on this slide a reminder up in the upper right corner that our objective as statisticians at this stage in designing your study is to decrease variability. Stratification does this by, at baseline, separating your sample by baseline characteristics such that the variability of the outcome is lower in each group than it is in the combined group. So based on our learnings in earlier phase studies or from subject matter experts, we learned that the stage of the disease, early or late, and whether the patient has received prior therapy, those are characteristics that will reduce variability of the outcome. So in each of these 4 cells, variability of the outcome is lower than if we had them all combined. Another design feature is enrichment. Enrichment is very much like stratification in that at baseline, we divide up the sample into different groups. The difference is that in enrichment, we drop one of the groups from consideration in our design. And this is common with genetic biomarkers, but it doesn't have to be genetic biomarkers. It's just some baseline characteristic. What we're looking for is a subpopulation defined by a biomarker or other baseline characteristics such that either the response to your treatment in one of the groups or the probability of the patients in that group having an event of interest is higher than in the combined group. So we say that the group is enriched so that the response will be higher. This is more common in the early phases of study, phase 1 or 2, because sponsor, regulatory authorities, everyone is interested to see if there is any cause and effect relationship with your product at all. And it might be difficult to detect if we don't enrich the sample, if we just look at the sample combined for those that may respond or not. We're working on a design right now in mild cognitive impairment, a disease that progresses rather slowly. It's very hard to measure, and we are looking and we're talking to the subject matter experts of how we might be able to enrich that sample early on and study it in the early phases. We're in phase 2 in the enriched sample, and then maybe in the phase 3 later on, we put the non-enriched sample back in place and study it together. That's what this slide is showing, is that in the phase 3, the later phases, you might decide to study both groups rather than just the enriched group. But again, that enriched group reduces variability, usually quite dramatically. Okay, another feature is, this one's a bit more complicated. This is called adaptive randomization. This is not just an adaptive design. It's adaptive randomization. Randomization is when you allocate a newly recruited subject or patient to one of the treatment arms, and you do this randomly. Adaptive randomization says you're not going to do that with equal probability throughout the study. You're going to adapt the probabilities at which a patient is assigned to one treatment arm versus another. And you're going to do that based on the recruited patients that come in to date. So I recruit 10 patients, I look at their outcome based on the results of those 10 patients. I might change the randomization such that the probability going to the higher dose is higher than it is to the lower dose. So they're no longer equal probability. This is common in oncology. Well, I shouldn't say it's common. It's becoming more common in oncology where we are concerned both about toxicity and efficacy. And what we do is we survey the subject matter experts. They tell us what they expect to see for the different dose levels of the treatment. On the left, you see a toxicity level increasing, and you see that dose 3, the highest level, is likely to be the most toxic. If you go over to the other chart, that high dose is also most likely to be the most efficacious. So we have to balance toxicity and efficacy. And this adaptive randomization measures for the early recruited patients, what is the toxicity, what is the efficacy, and the next patients that are recruited are more likely to receive a dose that is both efficacious and lower toxicity. This is an example of what I was calling a Bayesian system, that triangle off to the right, because we're not intervening this after each patient. We're collecting the data, we have our models, they're updated automatically. The allocation probabilities are determined automatically based on the data that's come in to date. And this system learns, and it will gear more patients as they are recruited to the safe and effective doses much more quickly than if we say we're going to recruit 50 patients, stop, look at the data, then decide how to continue, stop after 50 more, so forth. This is done patient by patient. So it is a true system. This slide is showing a design that McDougall came up with some years ago that incorporates all three of those features: stratification, enrichment, and the adaptive randomization. And this passed FDA scrutiny. FDA, in my experience, is interested in seeing complex, innovative designs if they are well thought out, well justified, and they are assured of good execution on that study. In this study, we had a kind of a lead-in phase, still an enrichment based on a biomarker that indicated whether a patient would not respond to the primary treatment. And if they were likely to not respond to that treatment, we would move them on to our investigational product. It saves a lot of time. Instead of waiting for the patient to receive the prior treatment, only finding out after 16 weeks or more that that patient didn't respond, and then enrolling them in our study. The biomarker could do that much more rapidly. After we reached a minimum sample size for each dose, then we triggered adaptive randomization. And this was done in a stratified manner for different baseline characteristics. And this is also a seamless phase 2-3 design that after either adaptive randomization automatically selected the proper dose, or we hit our maximum sample size and we chose our proper dose, then we did the phase 3 with just the control and that selected dose. A very, very common adaptation in a study is sample size re-estimation. Most of the studies that we design these days includes at least one sample size re-estimation. There's often uncertainty in the assumptions that are required in the power analysis that we decide a third or halfway through a study, we're going to stop. We're going to check those assumptions again, see if they in fact do hold. If not, we might adjust our sample size up within a certain maximum and then continue the study. We might decide to continue as originally planned, or we might declare that it's futile and we should stop the study. This is one of the most common adaptations. Okay, some more, what we'll talk about from this point forward. These are more recent advancements in study designs. Excuse me. Baskets and umbrellas, they were developed in oncology. They could be applied elsewhere. It's really quite a clever idea. This slide shows what would be done without the, well, I guess I'm calling this traditional designs. The shapes of the icons on the screen, square, circle, diamond, and triangle, are different cancer types, tumors A through D. And underlying these different cancers, there's different mutations. And the mutations can occur in any of the 4 cancer types. So the mutations are color-coded. And traditionally, what we would do is if drug 1, the sponsor is interested in checking against cancer type A, then we conduct a study in just that cancer. And the same for the other drugs. Or it could be that drug 1 maybe is useful in any of the 4 cancer types. Under traditional designs, we would have to conduct 4 different clinical trials to test that drug against those different cancer types or tumor types. What a basket trial says is that let's group all of the cancer types together based on the mutation type. Now, typically these oncology drugs will work theoretically based on the mechanism of action because of the underlying mutation in the cell. So all of the blue mutations are brought together across all 4 cancer types, and that drug is tested against all 4 cancer types in one single clinical trial. It's a way of saving 3 additional clinical trials. The research question changes slightly. Instead of, is there efficacy of this drug in this cancer? The question is, does this drug have efficacy in at least one of these cancers? And that slight change in the research question allows greater efficiency and allows the data across the cancer types to be pooled together. And if drug 2, it's intended to work based on mutation, the green mutation, then you could conduct a trial, again, combining the cancer types that have that mutation. Highly efficient clinical trials. The umbrella trials are similar, but it's from a different angle. If there are multiple drugs that work on a cancer type, then let's test all 3 drugs on that one cancer in a single study. So the question becomes, do at least one of these drugs show efficacy in a given cancer? I've heard of these trials being done that requires sponsors of the different drugs, the companies of different drugs, to hammer out some legal agreements on how they're going to conduct these studies. but it may be the only way to conduct research in rare diseases. It requires a lot of collaboration. Okay, platforms trials. These are more recent. Before I describe platform trials, let's look at a traditional stratified design and how we might adapt that design by dropping arms. Suppose we have two standard of care A and B, and then we have a treatment that we think might work with either one of those standard of cares. So we have standard of care A alone, B alone, and then with treatment. So 4 arms in the study. We decide maybe at 33% or halfway through, we'll conduct an interim. And based on some pre-specified criteria, we will drop arms that are underperforming. So this slide is showing that we dropped the standard of care B arms. So a few points to note. All populations are studied from the beginning. There's 4 treatment arms. We can drop the underperforming arms, and we might narrow the target population by enrichment. That is to say, we could include some of our other design features out of our toolbox for these studies. And we confirm or change the sample size at the interim, even for the 2 arms that are continuing on. Now, how does a platform trial differ? Platform trials are conducted by first writing a master protocol. This master protocol lays out the study design requirements, no matter what drug is going to be studied or which subpopulation will be studied underneath this master protocol. Those different drugs or different treatment arms then become appendices to the master protocol, and it's in the appendix that we lay out the unique requirements or features of the study for that specific treatment. So on this slide, you see 3 appendices, 3 different treatments, A, B, and C. Some of the things that might change might be the eligibility requirements and so forth. So again, the master protocol might specify eligibility requirements that persist across all these treatments, how the randomization will be conducted, what are the common objectives, the common endpoints, the common hypotheses, and so forth. The appendices will lay out the unique features for a given treatment or population. Typical, it's to change the eligibility criteria or a standard of care. This picture is meant to show how this rolls out over time. Here we have standard of care A, B, and C, 3 treatments, A, B, and C, and we decide that treatment A is going to be studied first. And we launch in January the treatment A arm and a standard of care arm. And then in March, we decide we're going to launch treatment B, and it will also have its own standard of care arm. Now, it's possible, this is one of the efficiencies, depending on what's being studied, that you can pool the data for the standard of care. So the dark blue line is showing that our cumulative placebo control or standard of care arm is growing without our individual standard of care arm having to change its recruitment targets. At some point, we're going to conduct an interim or 2 and decide whether to keep an arm. And maybe it's we want to see a certain signal in treatment A standard compared to standard of care A before we would launch treatment C and standard of care C. So what we're doing is we're starting out with 1 treatment, only 2 arms, whereas in the traditional design, we started out with many more. We start off with all of them. So in this one, we start small and grow big based on the signal we're seeing in the early data versus starting big and then dropping arms and getting smaller as the study progresses. So in that respect, these are much more efficient. And to combine standard of care arms or Sometimes even the treatment arms can be combined. That requires Bayesian statistics to do that. So the master protocol, it offers a lot of flexibility. We start small and expand as the evidence accumulates and instead of starting big. Successful appendices are the ones that are carried on to phase 3. The appendices can be dropped and added much more flexibly than in traditional designs. Bayesian methods can be used to increase the efficiency by combining control arms or combining treatment arms either in whole or in part. And you might even leverage historic data to reduce the initial sample sizes. I'll talk more about that in a little bit. And you can adopt changes in the standard of care appendices as they are implemented. So these are eminently flexible. An example of a master protocol is the one that was put out by the World Health Organization to study COVID-19. McDougall is supporting 3 different COVID studies, therapeutics, and they all borrow elements from this master protocol. And for the idea behind the WHO is they wrote the main protocol, how they wanted evidence to be collected for therapeutics for COVID, and if sponsors of these products wanted to increase the speed with which their trial data are reviewed, it was in their interest to follow this master protocol. Okay, we're doing well on time. This is one of my favorite because I think this is one of the ways to be most efficient is to not reinvent the wheel. And we cannot reinvent the wheel by leveraging historic data. Historic data are any data that have been collected prior to the study that you are currently planning. That historic data can be data from your previous clinical trials. It can be data from electronic health records. It can be data from other studies. It could be data that was published in the literature. There's all different kinds of historic data. The idea is to not collect more data in your current study if the information can be gleaned from historic data. And this is often called borrowing. The key to success is to find historic data whose underlying population looks like the population you are about to study or are studying. And that's what on this slide they call congruency. If the populations are congruent, then you can add historic data to data that you collect in a control arm that we're showing in the lower right here. So you can augment your control data with historic data. You could replace your control data with historic data. And it is sometimes possible to do the same with the treatment arm. It depends on where those historic data come from. All of this has to be laid out in advance, pre-specified, convince the regulatory authorities it's done without bias. We worked on a clinical design, really wasn't a clinical design because the study was just about finished, but it was a single arm study. It was never intended for submission to a regulatory authority, but a new sponsor took over that study, decided they wanted to present the data to a regulatory authority. The regulatory authority came back and said, in order to really claim cause and effect, you need a control arm. There was no control arm. The study was about finished. So we floated the idea of using historic data from published literature. The regulatory authority, to my surprise, they did not say no. They said, tell us more. Give us the details of how you will conduct your literature review, how you're going to guard against publication bias, how you're going to ensure congruency, and so forth. So I count that as a great success that we're able to continue the discussions with the regulatory authority about really synthesizing a control arm for a study that did not have one. Okay, just a reminder, this is a team effort. Statisticians, in order to bring these ideas to your attention, and there are others, and when you begin to combine the different ideas, it creates a rather large toolbox to increase the efficiency of your design. We can only do that if we are part of the team. So again, it's in your best interest to get a statistician involved early on. And with that, Chris and Heather, I think I will stop there and thank everybody for listening. And then we can start with questions.
Excellent. Thank you, John. That was fantastic. For anyone that has questions, feel free to type them out in the Q&A section in Zoom, and then we'll just read them out. After the Q&A, we're going to do the prize draw. We've got 3 prizes, and we've got a fun way of actually picking the prize winner. So we'll go into that shortly thereafter.
Should I stop sharing, Chris?
You can leave your slides up, John, in case there's a question that you want.
I go back to them? Okay.
Yeah. So the first question's from Michael Daly, and I'll do my best to read this out. My limited experience is that CDER's acceptance of Bayesian approaches versus frequentist has been spotty. Unlike CDRH, first to formally accept adaptive Bayesian design and CBER. Is this still true?
In my experience, I would agree with that. It depends on which group you're talking to at the regulatory authority. It in part depends on their own experience with Bayesian methods. And it depends on when you talk to them. The earlier, the better, the greater your success will be. Do your homework, be well prepared, practice the challenges that you're going to face. And you should realize better success, but there's no guarantee. But just before the pandemic started, so this would have been early March 2020, I attended an FDA, ASA, the American Stats Association workshop on complex innovative designs. And the FDA has a program where they encourage sponsors to submit to the program your complex innovative design. And they selected several, and they worked with the sponsor to flesh out the final design. To me, that signals that the FDA is encouraging these more, call them modern methods, and many of them hinge on Bayesian methods. We also, with a sponsor here in Canada, we worked on a design, and we considered a Bayesian design, and we decided not to promote that design or present it to Health Canada. We had our reasons, but we went with a frequentist design, and the response from Health Canada was they rejected the frequentist design, saying you should be using the more modern Bayesian methods. So again, although we were disappointed to be kind of shot down that way, we were also celebrating that Bayesian methods are becoming more accepted.
Excellent. Prem Pal asks, how many interim analysis are allowed for a trial?
As many as you please, really, but you have to be careful why you're doing those interim analyses. The adaptive randomization you could think of as an interim analysis after each patient comes into the study. It's that flexible, but you have to be careful why you're doing the interim, whether you're doing it for futility, efficacy, so forth. It can impact the power not the power, but the type 1 error rates for the final analysis. So you want to do things in moderation.
Excellent. Michael Young is asking if we'd be able to share the slide deck with all the attendees after.
Sure. Yeah.
Excellent.
I'll do a PDF for you, Chris, and you and Heather, I guess, can distribute.
Perfect. And we're also going to be sharing the recording as well.
Recording.
So we'll have access to that as well. Idis Lima is asking if you are aware of some good databases to obtain historical data for oncology trials.
I will have to think about that one. So I'd like that person's contact information, and yeah, I have to think about that one because I think there are, but I can't remember what it is at the moment. I seem to recall reading, because I seem to recall an email coming across my desk that said, look at this new source of data. And I never did look at it, but I recall it happening. So I'll have to look into that one.
Yeah, no, it's a common question, not only with the clinical data, but imaging as well.
Okay.
So Idis will get back to you on that one. Andrew Skura is asking, he says, today, what are the chosen formats for data set submissions? And do the health regulators require that standard for all data submitted?
For the FDA requires the CDISC data sets. Now that's on the drug side. Device side still has no requirements. That's the FDA. I'm not sure whether maybe somebody else knows and can write the answer in the chat, whether the EMA requires the SDTM and ADAM. Health Canada does not yet have that requirement.
Okay. Michael Young asks a further question. What is the productivity of umbrella/basket trials? Rate of surfacing viable indication targets? Financially, are these types of trials able to reliably produce a positive ROI?
Yes, they are. If it's well thought out, it's the target. So when you're still not sure, you understand the mechanism of action of a product, investigational product, and your subject matter experts know where that mechanism of action will work against what mutations. So now I have to be careful. I'm a statistician. I'm not a biochemist and biologist, right? So we're on the periphery, on the very edge of my understanding, and I want to be careful not to say something wrong. But if those mutations occur in several cancers, then asking the question early on, does this product show a signal or efficacy in at least one cancer type? A basket study can answer that question very efficiently. And then you decide from that point forward whether to continue and focus on that cancer.
Excellent. Ramanan asks, is there a possibility to use historic trial data as the control arm data without any additional subjects recruited for the control arm?
It's possible, yes. Yeah.
Perfect.
Yeah, and that's the story that we floated in front of Health Canada, and they're interested to learn more. It's better to do that up front. Make it known to your regulatory authority what you intend to do and get their buy-in early on. But it is possible. And there are, and if this person contacts me directly, I can give a reference, and it shows a table of how historic data was used. It's kind of a survey of how historic data have been used successfully so far in getting product approved by the FDA, and how those historic data have been used. It's an interesting read.
Excellent. We have two answers from Sonia. Thank you, Sonia. Number one, depending on the type of cancer, she says Medidata has a large data set, and confirmed the FDA requires both SDTM and ADAM.
Yeah.
Thank you, Sonia. Rudresh is asking, for the studies with multiple interim analysis based on the new data, if the endpoint assessments are changed, how does Biostats handle it?
Can you read that again, Chris?
Yeah, for studies with multiple interim analysis based on the new data, if
Yes.
endpoint assessments are changed, how does biostats handle that?
If the endpoint assessments are changed. Okay, I'm not sure what's meant by that. If you've changed the endpoints or how you measure an endpoint. I don't think I can answer that with a question that fits every situation. So this would have to be a case by case.
Mhm.
What is it that you want to do and change at each interim? And I think that if I understand the question properly, the key is if you change the endpoints or change how you're measuring them, in order to do the analyses for which it was changed to, do you have the data required for the earlier subjects prior to the interim? That's what would have to be careful about.
Okay.
I think. But this, I think the best answer is that's a case-by-case assessment for the protocol design.
Okay. Yeah, and Rudrash, if you want to dig into this, by all means, feel free to email Heather and I after.
Yeah, I'd be happy to talk to you.
Yeah, and we can follow
about
up
that.
with you directly.
Sure.
Pearl Mensa asks, how do you take the renewed push for diversity into account in your existing models and with your new trial designs?
Diversity as far as, I guess, ethnicity and race.
Mm-hmm.
How do we take that into account? I'm not sure I understand the motivation for this. So I guess what we're getting at is if one or more groups are underrepresented in the sample, perhaps.
Correct, yeah.
Right, okay, so there's ways to reweight the data such that those that are in the sample of the underrepresented groups account for more information than their counterparts. So now, if you recall in my introduction, I said that in my USDA days, I learned about probabilities. And some of the methods are starting to find their way into clinical research. And this is one of the ways. So that's a possibility, is to reweight. That one is actually a difficult situation to correct once it's happened.
It's almost something you need to address at the very beginning, right?
That you should, yeah, yes. And that's in the study design, you could stratify by those groups and require sample sizes of a certain size for each of those groups to make sure you get the representation. And we did this for a client one time, and it was a post-marketing study, but they wanted to ensure that their results were representative of different populations, ethnicities, I believe, across Canada. And so we worked out a design where it required that we first had a reliable source for the proportions in the general population, their target population, made sure our sample proportions matched those, and how we did that. But that was at the design stage. That was not a fix afterwards.
Right.
Afterwards, then you're talking about a reweighting, and although that works, it increases variability because those weights factor into your variation estimates. And if you have just a couple from a group with very high weights, it could render the results so that the confidence intervals are so wide that you might not have any confidence in your results at all. That's a difficult situation to correct.
Excellent. Thank you, John. We've got one more question from Andrew Skura. Historical data can be problematic when combining with current trial collections. Can you give a quick list of the cost benefits when using that legacy data? Essentially, when do you make the choice to use it or not?
I think it's most commonly used. You kind of have to do that assessment up front. That's why I say you get your regulatory authority buy-in. Say I'm going to reduce my sample size in the control arm from 100 down to 20 because I'm going to get 80 from historic data. They have to, you want them to agree with that. And then the benefit is you save the cost of having to recruit 80 patients. Mm-hmm. But you should know up front that the benefit is secure through your conversations with all the stakeholders, including the regulatory authorities. Without that, it's a very risky venture to rely on historic data and find out only afterwards that it's unacceptable. And the key then, of course, is the congruency. But it's becoming more and more common to rely on, and some people call it external data, simply because it's external to their current clinical study. The best data, the historic data to use is that which comes from your own previous clinical studies. Right. Could be of that product, could be of a similar product. Where this is gaining the most ground is in pediatric studies. When the product is approved based on clinical trials in adults, and then the regulatory authority requires pediatric studies post-market, or maybe as another phase 3, you have a wealth of information. And you get your subject matter experts together and you use Bayesian methods and historic data to leverage the data you've already collected on the adults to suggest what's going on. That gives you a nice prior distribution for your pediatric study. And that should reduce the sample size requirements a great deal. And pediatric studies are difficult. Parents are reluctant to volunteer their children for some studies.
Definitely. Michael,
for good reason.
Sorry, right? Michael Daly asked one more question. Is only patient level data required to be included in Bayesian approaches?
Patient level data? No. And Bayesian methods are in these historic data, and the design we floated with the historic data to Health Canada, we had no patient level data. It would be from a systematic literature review. So we would just have the parameters. What you need in Bayesian statistics is that distribution. Mm-hmm. That distribution is just based on certain parameters, mean, variance, scale, what have you. You don't necessarily need patient level data to get those parameter estimates. Patient level data is by far the strongest data. So if you can get patient level data from previous studies, electronic health records, so forth, the better off you are. Mm-hmm. But it's not, doesn't have to be individual level data.
Perfect.
And actually, I'll say one more thing. Yep. If you have somebody that has individual level data, and for privacy reasons, they don't want to share that with you, then you just tell them what you need, calculate these numbers from your data, and send that to me. And that's all I need. So it can actually maintain privacy in certain situations.
Good point. That's it for questions. So I'll open it up one more time. If there are any other questions, just type them in. And if not, then we'll flip over to the draw and see who our 3 prize winners are.
Okay, see, I need to. There you go, Chris.
Well, let me just switch over here real quick. And we are using a very scientific method. It's called the spinner wheel. So we're going to do a draw for 2 $25 gift cards to Starbucks, and then a $100 gift card to Amazon. So let's go. Let's see who wins the 2 Starbucks cards first. Well, there you go. Pearl's our winner of a $25 Starbucks gift card. So Pearl, we're going to take you out of the list and try again for the second Starbucks gift card. There you go. Ramanand's our second winner. We'll remove Ramanand. Now for the grand prize $100 gift card to Amazon. Here we go. There it is, Letitia Vito. Thank you so much, everyone. For the prize winners, you'll be hearing from Heather and I either later today or tomorrow on how to get your gift card. Thank you, John, again, for a fantastic presentation. As we mentioned in the meeting, we'll be sharing the slide deck as well as the recording, so you'll all receive that within the next few days as well. Thank you, everyone. Bye for now.